Hello everyone, welcome back to another class on regeneration biology. In today's class, we will learn about the tissue 3D printing and organ culture in detail. So far we have seen the prospects of 3D printing and making of different matrices, etc. for inoculation of the cells. Now we will see how you do 3D printing and organ culture in practice. What are the types of printers used, etc. we will study. So if you look into an overview of 3D printing, this cartoon explains you in a nutshell that what 3D printing is all about. 3D bioprinting is a state-of-art approach to the design and processing aspects of organ culture. The major applications in the biomedical area, including repair of bone, soft tissues and organs, they are very much prevalent. The current limitations and the latest evolutions in bioprinting based strategies are also need to be examined. The existing results and new horizons are discussed to provide a picture that is useful to both senior and early stage researchers. As you can see here, this is the bioprinting and tissue engineering system, and you can see for cardiovascular issues, skin, bone cartilage, liver, urinary system, cornea, reproductive system, muscles, glands, dental, respiratory system, neural system, tendons, etc. Due to the limitation of time, we will not address each and every tissue. We will pick a few of them and we will try to understand how it is implemented, how it is done. So this is another schematic illustration of the steps that is required to generate bioprinted tissue structures. So the bioprinting process involves fabricating functional tissues and organs by integrating and assembling various biomaterials and bioactive molecules in 3D. So 3D, when you say, it is about X, Y and Z axis. That is the meaning of 3D. So bioprinting techniques provides reasonable control over both cellular and cell-laden constructs by mimicking a specified configuration and surface and structural properties, leading to steering their cell activity. So the workflow of bioprinting techniques commonly starts from medical image datasets such as magnetic resonance imaging or computed tomography CT scan, which provide macrostructure, information of organs and tissues, then the architecture of the 3D structures with high fidelity is achieved through the computer aided design. You may have heard about mechanical engineer people use this CAD. Short form, it is called computer aided design software. So in a nutshell, you can say different organs are there in a human body, and you can see how a 3D CAD model of kidney looks like and what is the manufacturing strategy, and then go for bioprinting and then maturation and culture of that organ and implant. So it starts with the image acquisition in the patient, and then you try to map the entire organ and come up with a 3D printing strategy and you implement it. So 3D bioprinting is successful use of bioprinting technology. It is attributed to the functionality of the resulting structure, which is defined based on the components, structural components, and also what is the bioprinting device used and cell-cell interaction. It depends on all these three factors. Say for example, if you are putting a very solid metal structure on a plastic substratum support device, then that metal structure strength is not going to be of any much use because it is attached onto a plastic wall or a plastic material. So if your structure is metal, it should be at least anchored onto a metal or equivalent. Cannot be weaker than that. So this is the idea you should have. Several techniques have been developed for accurate and controlled deposition of different biomaterials, creating intricate structures that mimic native tissues and organs. Based on the principle of operation, three commonly used 3D bioprinting techniques exist as of now, which are inkjet based, laser based, and extrusion based bioprinting. These are all the three approaches that is available as of now. 3D printers, inkjet based. Let us understand how and why they function. Inkjet based bioprinting operates on the same principle as the traditional inkjet printing, but instead of using ink, it uses bio-ink containing living cells. This technique involves precisely ejecting bio-ink droplets from a print head onto a substrate to form a specific 3D structures. It has to, because you have a scanned image available with the printer already, which you got it from a scanner. Inkjet based bioprinting is considered a non-contact technique that creates discrete droplets under pressure and precisely deposits them onto a substrate at desired locations under control, where it interacts between the droplets and substrates are created. The interactions has to be created between the droplets and the substrate so that it will gel together. Pressure pulses impact the fluid chamber by overcoming the surface tension of the bio-ink, triggering the droplet ejection. According to various actuator mechanisms, whether it is a thermal, piezoelectric, electrostatic, etc., it can be different actuations can be applied, causing the droplet to eject. Because you have ink, you have ready ink ready, but how, with what force, which direction you will push them is what is to be taken care of. Inkjet print heads are temporarily deformed either thermally or piezoelectrically to eject the droplets of various sizes during printing, depending upon what you are going to print on a given location. The thermal inkjet printer uses a heating pulse from a thermal actuator to eject the vapor bubble and ink droplets from the nozzle. This is another approach of using what should be the actuation method. However, it may have limitations regarding printing viscous bio-inks and retaining cellular viability during the printing process. Having said that, every tissue has got its own viscosity, its own tissue density. Accordingly, the ink is nothing but the cells itself quality or the viscosity also has to change. Laser based, another printing approach, ink based is done, now it is laser based bioprinting, uses a mechanism similar to that of inkjet printers, but using laser pulses to deposit cells and biomaterials onto substrates precisely. Remember, these so-called inks should have both substrate as well as the cells, not just the cells alone, not just the substrate alone. This technique creates a laser-induced pressure wave that propels cell-containing droplets from the donor slide to a receiving substrate, forming the desired pattern. 3D pattern, not 2D pattern. A pulsed laser beam is directed onto the interface between the target substrate and the absorbing layer. So this causes thermal volatilization and the formation of the microbubbles, which is part of the integral part of the 3D structure itself. The expansion of the microbubbles ejects bio-ink droplets. Initial bioprinting systems using laser-based processes were also called stereolithography or SLA. So the SLA bioprinting techniques utilizes visible light or ultraviolet to layer by layer photopolymerize a photosensitive solution in specific areas to generate a suitable structure. So this is the step-by-step mechanism of laser bioprinter. The laser beam's small size allows the SLA to produce cell patterns with intricate structures and achieve high level of resolution to submicron level. Printing resolution, submicron means below 1 micron, it can reach, because one cell's diameter is roughly around 4 to 5 micron. So definitely not more than 10 micron. So submicron is a great resolution to have. Digital light processing, also known as DLP, and SLA techniques use photosensitive materials but their implementation or the actual procedures can differ slightly. During the DLP process, a digital micrometer device is used to target light onto a region that cross-links all points related to each layer. So this is the overall concept of using a laser printer. So 3D printers, when you think about, and the extrusion method, that is the third method. Extrusion based bioprinting is one of the most common and versatile techniques used in bioprinting. It is very well used because it has got lots of advantages and benefits when you implement it. It involves the deposition of biomaterial ink continuously through a nozzle using mechanical force or pressure driven by a delivery system. It can be a gas pressure or any other vacuum pressure, such as air, a piston, or a screw. Some pressure is needed. Simple approach. The bio ink is carefully extruded layer by layer to print a desired structure, 3D structure. The pneumatic extrusion based bioprinting drive system is simple and primarily dependent on the pneumatic pressure, while the mechanical drive mechanism provides better spatial control. So both have to work hand in hand in order to get proper resolution. More than that, the mechanical techniques regulate the bio ink more directly than pneumatic systems, which rely on the delayed response of the compressed gas in the pneumatic systems. Nonetheless, we should understand that the pneumatic systems offer advantages when applying various types of viscosities. Like earlier we discussed, the viscosity of the ink can be a detrimental factor, but here extrusion method, because you are applying a pressure, it is no more subjected to the ink's viscosity, etc. So it allows different types of viscosities of bio inks by adjusting the valve's gate time and pressure. So it can adjust the valve. So for a given pressure, it can decide based on its viscosity how much volume has to be pushed in. So both pneumatic and mechanical techniques can print highly viscous bioinks through mechanical system that might offer superior spatial control. So as long as the organ is going to be resolving in an adequate resolution, then the extrusion based, which is pressure based approach is more handy. So the resolution of extrusion based bioprinting is typically influenced by the pressure, nozzle diameter, size, deposition rate, and material type. But still, one thing you need to know is that the achieved printing resolution is relatively low. That only laser printer can provide submicron level resolution, but this is relatively low, that is ranging from 200 to 1000 micron. Laser printer is giving submicron level 200 micron to 1 millimeter. 1000 micron means that is a limitation compared to other bioprinting techniques. This technique offers high resolution and includes multiple types of growth factors in the bio ink because its resolution is low, but it allows better or a higher viscous bio inks also it can tolerate. Moreover, its case of use, scalability, and wide range of applicable biomaterials make it a preferred technique for printing biomimetic 3D tissue constructs. Despite its low resolution, people still use the extrusion method. However, passing bio ink through the nozzle diameter may raise shear stress because it's a viscous solution. You have to push through the nozzle, you have to apply more pressure, leading to a decreased cell viability. Ink is there, but ink is nothing but cell. You apply too much pressure, ink may not or the cell may not stay healthy. This is a challenging point needing optimization and affects the printing speed and size of cell aggregates that can be printed. The most widely used technique for biomaterial extrusion in 3D bioprinting is fused deposition model, also known as FDM. It's a type of extrusion based technique. So the integration of 3D bioprinting with other approaches. The 3D printing offers a versatile approach of fabricating customized biomaterial scaffolds with interconnected pore networks that facilitate the transport of proteins, oxygen, and other nutrients compared to the traditional methods or traditional techniques. However, we should understand one more thing: many existing 3D printing methods lack the resolution to produce filaments suitable for scaffolds across various tissues. When the resolution is low, you cannot expect the ECM to be made as it should be, because cells are in and around 5-6 micron. So ECM also will be around that range. Additionally, the pore size of the 3D printed scaffolds are often larger than the size of the cells, adversely affecting cell seeding efficiency and tissue formation. Instead of a thick normal cell, it will look like porous, it will look like a sponge. You want to look like a brick, but now the tissue is looking like a sponge because the matrix was larger and cells are attaching, the middle is leaving empty. So this is the issue. Among these techniques, electrospinning stands out, a robust and straightforward method that employs high voltage to generate nanoscale fibers with a large specific surface area. So this is taken into consideration when you are using a 3D bioprinting with other approaches which you are catching on to. Combining 3D printing and electrospinning techniques proves to be advantageous to overcome several limitations. This integration of these two techniques, that is 3D printing and electrospinning, allows the creating materials with controlled shapes, highly porous interconnected structures, sufficient support strength, and the ability to incorporate nano patterning, which is very friendly for a matrix to be fabricated. It also provides ECM-like niches and bioactive cues to cells. Depending on the specific application, 3D printed scaffolds and electrode spun fibers can be combined into various ways, such as electrospinning onto 3D printed scaffolds, 3D printing onto electrode spun fibers, alternating the use of 3D printing and electrospinning. Means you can do this permutation combination using electrospun fibers as inks for 3D printing and decorating or infusing the 3D printed scaffolds with electrospun nanofiber segments, or fabricating the electrospun scaffolds onto 3D printed collectors or templates. Basically, mismatch either of them, just like you can jumble up so that you end up getting a structure which is quite friendly and usable. Now let us see some example 3D printing approaches to engineer skin tissue. It's a schematic illustration of the design strategy and applications of a 3D bioprinted photocross-linked hydrogel of SF gelatin for full thickness to fix a wound repair or skin wound. That is what you are seeing in panel A. In panel B, illustrations of the hydrogel treated incisional skin wounds at a specific time. Panel C, the simulation of the corresponding wound. You are creating a similar wound because you are going to fix it. These are all done in rat models or rodent models. Panel D is a quantitative analysis of the wound site progression rates in different groups, how it is successfully staying in different rodel models in a in situ bioprinted scenario. And E is the workflow. As you can see here, panel E is the workflow of an adaptive multi-degree of freedom in situ bioprinting. As you can see here, this is the workflow. And F is the generation of hair follicles, is an example. Generation of hair follicles within 4 weeks following the robotic bioprinting and manual implantation process. And in G, here is the total number of hair shafts found in each group and wound. And H is what you are seeing is the skin generated inside and outside. So this is the overall implementation of a skin that is generated through bioprinting approach. Then 3D bioprinting approaches to engineer cardiac tissue. It is not that easy. The heart is the first functional organ that is formed during embryogenesis. Although the tissue is neuronal tissue, the full-fledged organ is heart. And it has a stylized 4-chambered muscular anatomy and is responsible for continuous blood circulation throughout our body, which we all know. A complex hierarchy of cellular diversity consisting of cardiomyocytes, endothelial cells, fibroblasts, connective tissues, and smooth muscle cells, and other specialized cells which are present, they pose great challenges to the manufacturing of the functional heart tissue. Although the 3D bioprinting cardiac tissue is still in the early stages of exploration because it's a tough tissue, recent research has indicated the possibility of 3D bioprinting functional heart tissue, particularly customized cardiac valves and patches. However, such approaches have limitations regarding full vascularization and synchronized contractile activity. Although tissue is fine, it should contract perfectly. Setner et al., a researcher, he introduced a new generation of anatomically precise 3D functional patient-derived models at various stages that can be used as a high-fidelity and robust platform to study various cellular microenvironment interactions comprising the geometry, ECM composition, flow thermodynamic, flow hemodynamics, biomechanics under both dynamic and static flow conditions of the cardiac tissue. So models of human embryonic heart tube and fetal left ventricle were bioprinted using hydrogel based seeded with endothelial cells and analyzed experimentally and computationally. Based on the cardiac geometry and flow conditions, there was a great variation in the endothelial cell proliferation and endothelialization in the bioprinted constructs. So these results show that a similar and precise flow hemodynamic pattern in the 3D space and the bioprinted constructs exist. By focusing on optimizing bioprinted cardiac patches, Roche and colleagues developed alginate gelatin-based heart patches for myocardial regeneration. The results showed a presence of EC networks which formed a durable structure and exhibited the contractile function, the most important function of the heart. Polonchuk and colleagues successfully constructed functional cardiac spheroids composed of ECs, endothelial cells, fibroblasts, and human cardiomyocytes blended into optimal alginate gelatin hydrogel with viscoelastic properties comparable to native heart tissue. That is quite promising. As a result of spontaneous contraction and stimulation of bioprinted cardiac tissues, cardiac spheroids, it was possible to record electrical signals and contractile properties of this tissue on the microelectrode plates. So these results indicated that the bioprinted cardiac spheroid has a great potential to reconstitute human cardiac tissue for long-term in vitro studies because this much progress has been made. So now if you look into the cornea tissue, which we all know, when you say that donating the eye means you are donating basically the cornea. Cornea is a highly organized, dense, avascular and transparent tissue which protects eyes from the external environment and is responsible for incident light transmission and refraction. Not just a window, but it also refracts the light. It has a specific arrangement of collagen fibers forming 200 to 250 lamellae, that means layers and layers, which give strength and spherical shape. The tissue's collagen lamellae and anterior part is intertwined and aligned with the middle and the posterior regions. So the transmission and refraction of light, which then focuses on the retina, are caused by the particular alignment, the way in which they are arranged, different lamellae. They contribute to the functioning of the cornea. An important contributor to blindness globally is corneal disease. Currently, the corneal transplantation is the most common treatment for corneal blindness. We have to wait for a donor, which is normally from the cadavers. However, donations are the main source of corneal transplants, far from meeting the demand, because you are not able to supply even 10% of the demand. A critical shortage of donor corneas has prompted research into effective corneal replacements. So, nonetheless, the many questions remain that the current research cannot resolve, including the restoration of optical function and reconstruction of the original geometry of the eye. That is the real question. The 3D printing allows the deposition of materials on digital command to construct results with sophisticated geometric patterns that can be created because of the 3D printing. Advances in 3D bioprinting technology has expected to allow precise control of corneal curvature and thickness based on specific refractive requirements of individual patients. Cornea's property change from patient to patient, overcoming the drawback of traditional tissue engineering. So the two major challenges for conventional corneal materials are rapid epithelialization and stable epithelial processes, because you don't want the cornea to become opaque because of the epithelial or vasculature growing on them. These processes are attributed to the cornea's curvature, which determines the epithelial response to corneal damage and the corneal healing process. On a lighter note, we can know that we don't want blood vasculature to form. So for other tissues, we want angiogenesis. But in cornea, angiogenesis is a menace. It will make the cornea opaque. In a study by Zhu and colleagues, gel MA and collagen were utilized to generate smooth 3D printed convex corneal implants. These implants exhibited curved structures that could regulate cell organization and adhesion. So this is a 3D printing approach to engineer the cornea tissue. Cell proliferation, live and dead. You can see a schematic of the PEG-A, gel MA, hydrogel formation and bioprinted procedure in the panel A. In the panel B, there is a cell proliferation, live and dead image, that is change in the transparency of RCEC containing hydrogels, and that you can see in this panel B, and this is panel C is done in the rodent model. In vivo evaluated the effectiveness of the corneal scaffold in a rabbit model. It's a rabbit model, not rodent model, rabbit model, and a graduated reduction in the size of the corneal epithelial defect represented by the green area, what you can see here, mentioned here. Green area, center of which is the center of the cornea, was observed due to the migration of the epithelium to the hydrogel and the defect. So in the panel D, what you can see here is the gene expression and the histological analysis that is shown by red arrows, and the rectangles that represent unfinished regions of the epithelium and the stroma. In panel E, the thickness of the entire layer epithelium and the stroma of the cornea, and relative gene expression of different genes, LDH, AQP1, these are all specific to the cornea, in the cornea, and that is confirming that the cornea is being made in a healthy manner. So, considering this progress, like the values reflect the fold change, as you can see here, these approaches, they give lots of promises, and we can conclude in some way that the powerful capability of 3D bioprinting will make it possible and practicable to streamline manufacturing processes in future. Because like I told you, due to the limitation of time, I am covering only few of the organs like kidney, heart or cornea. There are many other tissues also can be worked out. Advances in technological processes, computational modeling, artificial intelligence and machine learning, as well as a more comprehensive understanding of the optimal interactions between the biomaterials and the cells will significantly improve biomimetic capabilities of bioprinted tissues in the coming years. Bioprinting can be applied to create complex biomimetic tissues, allowing for the inclusion of vascular, lymphatic, and neural networks. So what we have learned so far is the promises that the 3D printing is offering to fix damaged organs in patients. We will learn more about the regeneration biology in the next class. Thank you.